Okay, so we are going to start talking about carbohydrates. So we're talking about metabolism, and we're going to start talking about the catabolic process called glycolysis. Glycolysis is the first step of cellular respiration. And it's glyco means glucose, lysis means breaking apart. So it's a 10-step catabolic process. And it breaks down one molecule of 6-carbon glucose. So it's 1,6-carbon glucose to 2 3-carbon molecules called pyruvate. You'll also hear pyruvate called pyruvic acid sometimes. It just depends on the pH. It's the same thing. So this is a catabolic process because we're going from something that's bigger to 2 molecules of something that's smaller. Before we get into glycolysis, we need to take a little bit of time to understand these molecules called carbohydrates. So carbohydrates have a lot of functions in biology and biochemistry. We are mainly going to be talking about them as a source of biological energy. We're going to talk about them in the first step of cellular respiration. They also are really important in the immune system, recognizing antigens and antibodies, cellular protection. They're important in bacteria, plant cell walls, cell signaling, adhesion, biological lubricants, protein trafficking, cellular structure. They have a lot of really important roles, and we will mostly just be talking about carbohydrates as an energy source, but there are many functions for carbohydrates, and we're just focusing on one of those. So carbohydrates, just from the name, carbo means carbon, hydrate means water, which is hydrogen and oxygen. So carbohydrates are made up of carbon, hydrogen, and oxygen. And then there are different types of carbohydrates that can be modified to include nitrogens or phosphates or other groups like that. But usually a carbohydrate, just a simple carbohydrate, is made up of carbons, oxygens, and hydrogens. So there's a lot of slides, a lot of good information about carbohydrates. One of the slides, slide 3 from this lecture, starts to talk about how we name carbohydrates. Carbohydrates have the suffix ose, O-S-E, not to be confused with enzymes that have the suffix ase. So ASE means it's an enzyme, OSE means it's a sugar. And oftentimes we will name sugars based on the number of carbons and the location of the carbonyl group. So there's 2 major types of sugars. There's what are called, so for example, there's aldoses and ketoses. So an aldose is a sugar where the carbonyl group is not on the end of the carbon chain, so it essentially has an aldehyde. And a ketose is a sugar where the carbonyl group is, sorry, I think I just said that backwards. The aldehyde is where the carbonyl group is on the end, a terminal carbon, and then the ketose has a ketone where the carbonyl group is not at the end. So there's lots of examples of this, and you'll see the names of sugars. We're not going to worry too much about the nomenclature and the systematic naming. You should be able to recognize the difference between an aldose and a ketose and that sort of thing. So when we talked about amino acids, we said all amino acids except for glycine were chiral. So sugars are going to have multiple chiral centers. So you can have multiple chiral centers in a sugar depending on what kind of sugar it is, which means you can also have L and D forms of sugars. When we're talking about glucose, we are mostly talking about D-glucose. It is not the case that we only use D sugars, just like we only use L amino acids, but that's just the form that we will be talking about most frequently when we are talking about carbohydrates and glucose going into glycolysis. There are different projections and ways to draw carbohydrates, either in a linear form or in a cyclic form. I am not going to be requiring you to draw those or to do any of those kind of projections, chair or boat conformations. We're not going to worry about a lot of that. We are going to talk about the chemical reactions that are happening in glycolysis. But this is just to kind of get us oriented to what's happening, what carbohydrates are. So let's see. So when we're talking about carbohydrates, if you remember way back to the very first topic, we said there are 4 different classes of macromolecules: carbohydrates, proteins, lipids, and nucleic acids. We said they all have a monomer form, a polymer form, and then the type of bond that holds them together. So for carbohydrates, the monomer is called a monosaccharide. So saccharide means sugar. Glyco also means sugar. There's a few different words that we use to represent sugar in biology and biochemistry. The polymer is called a polysaccharide. And the bond is called a glycosidic bond. The place that the glycosidic bond is first introduced in your lecture is on slide number 13, and this is a condensation reaction. So this glycosidic bond is a condensation reaction, similar to how a polypeptide is formed, but the peptide bond is a condensation reaction. And it forms this glycosidic bond between 2 individual monosaccharides to form either a disaccharide or a trisaccharide. So a water is removed, and then we form what's called a glycosidic bond. So this formation, this condensation reaction, the opposite of that would be hydrolysis, where we were breaking a glycosidic bond. That would be controlled by enzymes. So then there are some examples of common dimers, disaccharides, and their connections. So you'll see that there are what are called alpha glycosidic bonds or beta glycosidic bonds. And that has to do with the side of the ring structure that the hydrogens are on. You'll see this when you look at the picture. If they're on opposite sides or the same side, will tell you if you have an alpha linkage or a beta linkage. And the alpha or the beta linkage is important because when you're cleaving a glycosidic bond, enzymes are specific to alpha linkages or beta linkages. So special enzymes will cleave either an alpha glycosidic bond or a beta glycosidic bond, but usually not both. So there's all types of different names for polysaccharides. When we were talking about proteins, there was just a protein. We called it a polypeptide. But there's all different ways that these polysaccharides can come together, can have different branching, different types of linkages. And that there's a table in your book and in the PowerPoint slide, it's on slide number 18, where it talks about all the different types of polysaccharide. So there's starches. Starches are how plants store carbohydrates. I'm just going to write polysaccharides. So starch is how plants store carbohydrates. Glycogen is, or another way that plants store carbohydrates is cellulose. Also plants. Animal cells like us and also bacteria, we store carbohydrates as something called glycogen. And there are all kinds, there's many other types of polysaccharides. These are the three that we are going to talk about. So starch and cellulose are how plants store carbohydrates. So all plants are made up of some starch and some cellulose. So the deal is that our animal cells, not all animals, but humans, we lack the enzyme. Starch uses an alpha linkage, alpha 1,4 or alpha 1,6. Cellulose, when it is stored, uses a beta 1,4. So animals, I'll just say humans, not all animals, lack the enzyme to break the beta 1,4 linkage. Which means when the glucose gets stored as cellulose, we can't break those individual glucose molecules apart and use them to go into glycolysis. This is why plants that have a high cellulose composition have no calories or very few calories, because remember, calories are just a unit of energy. It's just a measurement of energy. If we can't get any energy from this molecule, then it would have no calories for us. So vegetables like celery is high in cellulose. So we don't use this cellulose for any energy. What we do use cellulose for is dietary fiber. So it's really important that we eat a lot of fruits and vegetables that are high in cellulose, but not because we're getting any energy from them. We're actually using it for dietary fiber. So plants like celery, lettuces, leafy greens, that sort of stuff that's high in cellulose has very low calories because we're not actually able to break that cellulose apart and get the individual glucose molecules for us to make energy. Starchy plants like potatoes or fruits like bananas or apples, they store their glucose as starch. And we do have the enzyme. We do have the enzyme to break the alpha linkage. So for example, a potato has stored all of these glucose molecules as starch with an alpha linkage. We can break those starch molecules apart into individual glucose molecules and then put that glucose straight into glycolysis to make energy in the form of ATP. So plants can store carbohydrates in two ways, as starch or cellulose. Starch provides energy for us because we have the enzymes to break that alpha linkage. Cellulose does not provide energy to us because we lack the enzyme that breaks the beta linkage. Glycogen, how we store glucose, also has alpha linkages. So we store any extra glucose that we take in, we store as glycogen. We'll talk about all of this later when we get into glycolysis and metabolism and a few more lectures. We store it as glycogen, but there's a limited space for storing glycogen, and once those glycogen stores are full, then any excess glucose gets stored as fat. So glycogen is how we store glucose in our cells. Starch and cellulose is how plants store glucose. And depending on the type of linkage, if it's starch or cellulose, we may or may not be able to use that for energy. Let's see. So there's a lot of really good overviews of what are coming up next. I've used some slides from a few different textbooks. And we'll get into glycolysis and then look at the 10 steps that takes this glucose molecule into 2 3-carbon pyruvate molecules.